Hi, I'm Peter the Pipe Cleaner. Today we're going to be explaining mitosis and meiosis using pipe cleaners. The uniqueness of using pipe cleaners is it allows us to color coordinate and intertwine easily to use chromosomes. Mitosis and meiosis are very important processes for cells. Without them, we would not be able to replace old cells, reproduce, and virtually we would go extinct. So let's begin. I have Jess and Mariah here to help me out with this.
In the first step of mitosis, unduplicated chromosomes become duplicated during synthesis phase and interphase.
Following the G2 phase, cells enter the mitotic phase. The nuclear envelope breaks down and chromosomes begin to condense in prophase. Here is prophase.
Then, chromosomes attach to the meiotic spindle from both ends and align at the center of the cell during metaphase. In anaphase, the sister chromatids of the duplicated chromosomes are pulled apart, and now independent chromosomes are then brought to opposite sides of the cell.
In telophase, the nuclear envelope begins to reform and chromosomes begin to reform. The cell prepares for cytokinesis, where the cell splits to result in two identical daughter cells that are diploid.
Now let's do meiosis. Red will symbolize maternal and yellow will symbolize paternal, just like it did in mitosis.
Meiosis 1 starts with unduplicated chromosomes becoming duplicated during synthesis phase and interphase.
Next, the cell enters the mitotic phase. The nuclear envelope breaks down and chromosomes begin to condense in prophase 1. Maternal and paternal homologous chromosomes join together to form bivalents, as shown here. These bivalents have genetic crossing over, which happens between non-sister chromatids. Centromeres also begin to form the spindle apparatus.
In metaphase 1, the homologous chromosomes align at the center of the cell. The way the homologs align at the center of the cell has to do with the independent assortment of alleles and the probability of the gametes carrying each allele for a certain trait. Additionally, both alleles have the same chance of being in one particular sperm cell as the other, as random segregation suggests so.
During anaphase 1, the homologous chromosomes are pulled apart and taken to opposite ends of the cells. At the end of telophase 1, homologous chromosomes then form new haploid cells, now that the cells contain half the number of chromosomes than before they were diploid.
Now that it is split into two cells, I'll get us started with meiosis 2. In prophase 2, the chromosomes recondense while the nuclear envelope breaks down and the spindle begins to form.
In metaphase 2, the chromosomes line up at the center of the cell again while connected to the spindle apparatus.
Following metaphase 2, the recombinant chromatids are split, forming independent chromosomes. The chromosomes are taken to opposite ends of the cell.
Finally, in telophase 2, the nuclear envelope reforms in all the haploid cells. The result of telophase 2 is 4 gametes with unique genetic information from both parents. Those 4 right there. Now that we've explained mitosis and meiosis, I'll let my friend Joel explain the genetics problem.
Our genetics problem has to do with 2 traits: the ability to tongue roll and to taste phenylthiocarbamate, or PTC. Tongue rolling is dominant to non-tongue rolling, and PTC tasting is dominant to non-PTC tasting as well. In our problem, a homozygous tongue rolling and non-PTC tasting woman and a heterozygous tongue roller and PTC tasting have 3 children. The question is whether the man is heterozygous for the PTC tasting trait or not, and to figure that out, we must look at the offspring and make dihaprid crosses with each possible genotype of the man. One heterozygous and the other homozygous for the PTC tasting. First off, we tested the cross if the man was homozygous and found that all the offspring exhibited tongue rolling and PTC tasting, which contradicts the phenotypes of one of the offspring that is non-PTC tasting, therefore meaning the man can't be homozygous for PTC tasting and must be heterozygous. To further prove our answer, we tested the heterozygous PTC tasting cross with the woman and discovered that all phenotypes that came as a result for the cross correlate to the phenotypes of the children. Half of the offspring would have the trait for PTC tasting, whereas the other half would not, which strengthens our answer.